Thank you. I'm really happy to be here, and yeah, it's nice to follow David. Obviously, impossible to to match that, but you know, I'll give it my best. Actually, yeah. So we're trying to use some of the tools that I learned in David's lab at Scripps and IAVI, and working with Dennis Burton and colleagues to merge that expertise in antibodies and knowledge of viruses, trying to merge those two fields to design new kinds of vaccines for vaccine targets that have evaded the classical vaccine strategies. And so the first slide is just, actually there's a website from the FDA which you can just download the list of all the licensed vaccines in the United States, and you see that all of these vaccines fall into these this small set of categories that have been known and done for a very long time: live virus, live attenuated virus, whole inactivated virus, subunit vaccines, and various pieces of bacteria. And these vaccines are remarkably successful. They're the most important tool for preventing an infectious disease in the human population, and they probably have saved more lives than any other feature of medical science. But there's still a lot of work to be done. There was a paper published in The Lancet in 2012, a series of papers on the causes of death and years of lost life in global health. And I took out this one figure that I don't expect you to read all of it, but it's showing for men and for women from, this is ages 15 to 49, in color is showing all the different causes of death. Actually, sorry, for each column is a different age group. So from all the age groups from 0 all the way to the end of life, and in color is marching out all the different causes of death in the year 2010. And if you look at the statistics, and I don't expect you to do it here, but you see that there are all these infectious diseases that are causing, on average, about 18.5% of all the deaths in the world in 2010. And this 18.5% we've looked at as caused by diseases that we think are potentially vaccine preventable. For some of them, the vaccine already exists and it's just not making it to the people that need to get it. But for many of them, we need new or improved vaccines. And I've listed down here some of the major ones that would make a gigantic impact on global health: HIV, malaria, tuberculosis, influenza, dengue, RSV. And in the talk, and sort of in my lab, we focus most of our time on HIV. And I'm going to tell you mostly about HIV. But we're also working on influenza and RSV. And I'm going to tell you a case study from RSV here that we think informs our methods for designing vaccines. And the overall idea is that those standard methods for designing vaccines have not worked for these major targets, and we need new strategies. And I'm going to, in telling you about the structural biology of HIV at first, I'll try to convey that one of the major problems is in focusing the immune response onto key epitopes, and we'll get to that. So HIV, it infects, this is a cartoon of a virion covered with glycoprotein spikes that bind to CD4, and that also, after binding CD4, bind the co-receptor, and after those two events, a series of conformational changes ensue, and the viral membrane fuses with the target cell, and the RNA goes into the host cell, and the cell is infected, starts producing virus. And for a vaccine, we need to induce antibodies that prevent that process, that neutralize the virus. This is a cryo-EM structure, a newly solved one that was just published in Science about a month ago, of the HIV trimeric spike. And GP41 is down here, so this would be the viral membrane, and this is the top of the spike. The CD4 binding site is in yellow, so this is where the trimeric spike engages the cell surface receptor, and so one place to try to induce antibodies to block infection of target cells is to induce antibodies against that yellow patch. But the trimeric spike in HIV is not naked like you see it here. It's actually covered in glycans, and we've modeled almost all the glycans in this movie. These things have lots and lots of degrees of freedom, and we actually built a computational protocol to simulate what they might be doing in solution. And you can appreciate that the spike is basically covered in these glycans, and they, because they're decorated by the human cells that the virus has infected, they look like self. And so HIV, and being a persistent virus that's been around the human population for quite a long time, at least 50 years or more, has evolved this trick to evade the immune system. And our challenge is to design a vaccine that will induce antibodies that can bypass this glycan shield and target conserved epitopes on the spike. One of the really daunting challenges, aside from that glycosylation about HIV, is the sequence diversity, and most of it occurs at the top of the spike that I showed you just now in the variable loops. This slide is taken from a paper by Betty Korber in 2001, and it's showing 3 different panels, and the distance inside the panel reflects number of mutations between 2 different sequences, so sequence diversity. And you can see on the left, this is basically the sequence diversity of influenza HA in 1 season. And in the middle, this is the sequence diversity of the HIV envelope protein in a single individual. And you can appreciate that the expanse of sequence diversity here is about the same as here. And then on the right is the sequence diversity of HIV in 1 country in 1 year. And you can see that the sequence diversity is vastly in excess of what you run into for influenza. So this slide is a really good way to think about, we all know that we have to take an influenza vaccine every year just to protect against seasonal flu. And so in vaccine world, we think of that as it doesn't work very well. We'd like to have a universal vaccine. You can imagine trying to do that same kind of strategy with HIV. It's totally impossible. There's 30 million different strains circulating, and when you get infected, HIV further mutates and escapes the neutralizing antibodies. So this slide, you don't have to look at the details of this slide, but this is from a study from Doug Richman in 2003, where he had a cohort of high-risk individuals, and he monitored them right when they got infected and then for months and months after they got infected. And he took samples of the virus and he took samples of the plasma, and he could measure in an in vitro neutralization assay, could the plasma at a certain time neutralize the virus at that same time. And what he found was, if you look along the if you look along the diagonal is asking, does the plasma neutralize the contemporaneous virus? And the neutralization titers are very low. You can see when you get above the diagonal, like here, the 6-month plasma is pretty good at neutralizing the day 0 virus. But the 6-month plasma doesn't neutralize the 6-month virus very well at all. This is the virus staying ahead of the immune system. And so we have to deal with that in a vaccine. Fortunately, from work, a lot of the work from Dennis Burton and colleagues, we have human antibodies that have been discovered that are broadly neutralizing against HIV. That means they potently neutralize the virus, not just one strain of the virus, but many, many different strains. And this schematic is just illustrating that basically there are 4 different sites where these broadly neutralizing antibodies have been mapped to bind to the spike. So similar picture of the trimeric spike here with the blue purplish glycans, and then there's one class of antibodies that's sort of coming straight into the slide, and that's binding to the yellow CD4 binding site that I showed you before. There's a class of antibodies that bind down at the base to the membrane proximal region, and then there are sort of 2 different major epitopes at the top of the spike, one right at the apex basically, which is binding to V2 glycans, and then one a little bit off to the side, which is sort of a V3, V4. And we have crystal structures of all of those many of those antibodies in complex with their epitope. This slide is just illustrating these VRC01 class antibodies that bind to the CD4 binding site. And they're pretty interesting because they use most of their contact, they use the heavy chain, a VH12*O2 heavy chain, to make their contact to the yellow CD4 binding site. And we'll come back to this. But then the classes of antibodies that bind to the top of the spike are very unusual in that they contact both protein and glycan. In fact, this is a crystal structure of an engineered protein that we designed, onto which Ian Wilson's lab added a mini V3 loop. And then Ian's lab solved this crystal structure of this antibody PGT128, which is incredibly broad and potent in neutralizing HIV. And you can see the CDRs reach in and they intimately contact these two glycans, as well as grabbing protein residues on the V3 loop. And so these are the kind of sort of strange antibodies that we need to be able to elicit. And so I talked about the, right in the first slide, the standard methods, so the classical strategies for vaccine design, and those are not going to work for HIV, and so far they haven't worked to design a universal flu vaccine. There are many other vaccines we'd like to design that they haven't worked for, but Dennis back in 2002 suggested the idea that's illustrated on this slide, which is that if you have an infectious disease and you have infected individuals, you can try to isolate neutralizing antibodies, protective antibodies from those individuals, and then characterize the interaction of the target envelope or other antigens that those antibodies bind to, and then use that structural information to try to design immunogens that could, the idea would be you design a cocktail of immunogens and then you vaccinate an individual and you would protect them because they would produce similar broadly neutralizing antibodies. So the nice thing about this strategy is you start with an antibody. This harkens to the discussion we had yesterday in the sequencing workshop. You start with antibodies that you know humans can actually make. It's part of they're part of the repertoire, although in the case of HIV they can be very mutated and strange. So I showed you that movie and of the trimeric spike, and that movie is of a real structure of an engineered trimer, and this, as I mentioned, these have been recently published, and that was a major advance in our field because we hadn't before had a recombinant molecule that was an accurate antigenic and structural mimic of a functional spike. And so now we do, and these papers have just come out, and that's a very, very attractive starting point for vaccine engineering. But I just want to show in this slide the degree of difficulty that we're going to encounter in using, even if we have a nice stable trimer. So what I have in this slide are side view and a top view of an unliganded spike, and I'll just start describing this here. It's the same, it's a frame taken from the movie that I showed you, so the glycans are this purple color. In yellow, I've colored residues on the protein surface that are exposed and highly variable across HIV strains. So you can appreciate that if you take one stable trimeric spike and immunize with it, you are likely to get a lot of antibodies that are very, very strain specific, and that's exactly what happens in natural infection. When a person gets infected, they make antibodies that do neutralize the strain that came in the door, but they're not broadly neutralizing. And this is not useful to us for a vaccine. And then on the panels that go over to the right, I'm showing you models for how these well-known broadly neutralizing antibodies interact with the spike. So this is PG16 that binds right to the apex. And you can, and then here's 3 copies of PGT128 and 3 copies of PGT122. All of these are very potent and very broadly neutralizing. But what I'm trying to show in this slide is that they bind to very precise conserved epitopes that are incredibly closely intermixed physically in space with highly variable residues. And it is amazing that the human immune system figured out how to make these antibodies be so broadly cross-reactive given the degree of variability that they're reaching past. And so our challenge in vaccine engineering is to figure out how to induce very precisely these kinds of specificities. And so that's sort of an argument, and it's recapitulated on this slide. I don't want to go through too many more details, but you can imagine there's a whole effort of trying to engineer trimers to focus responses on the conserved epitopes and away from those type-specific epitopes. And I'm not going to go through the trimer engineering strategies today because that's a whole long talk, but one idea is, could you combine a trimer and a heterologous prime boost immunization with a precise epitope mimic? So you say in an epitope mimic, it doesn't contain anything on the trimer except the conserved epitope that you want. You prime those B cells and then you boost, say, with a trimer. And so we're really thinking that that sort of strategy may be may be essential. And there are other issues here. Several of those antibodies in the last slide, they're all binding, making very extensive contact to glycans, but these glycan-dependent epitopes are likely to be very poorly immunogenic. So we may need special efforts to prime responses against those epitopes. The CD4 binding site, that yellow patch in the movie, is recessed. It's in a little cave, and then there's glycans flickering all around. So you can imagine, if you immunize with a trimer, you may never get antibodies against the CD4 binding site. It could be totally immunorecessive. And so we may need to prime with something that shows that yellow patch without anything else, and then boost with a trimer. So really, I think about it a lot as a problem of immunofocusing. And the rest of the talk is going to be two different stories of immunofocusing. And the first is a proof of principle for structure-based vaccine design, in which we've presented an epitope for an RSV neutralizing antibody on small minimal scaffold proteins using kind of strategies that David just told you about. So respiratory syncytial virus is also a really important global vaccine target. Probably everyone in the room has had RSV at least once, if not several times. Outside the developed world, it actually kills about 6.5% of kids from the ages of 1 month to 1 year. And so a vaccine is really seriously needed. There is an FDA-licensed prophylactic antibody, Synagis. I'm sure you're all familiar with that. And it makes about $1 billion a year. And so the idea, we sought to try to design a vaccine that could elicit similar reactivity. And our colleague Peter Kwong and Jason McClellan at the VRC solved the crystal structure of motivizumab, which is an affinity matured version of Synagis, with its epitope from RSV, the RSVF protein. And so motivizumab is in surface here, and the epitope from the end to the C terminus is the helix-turn-helix motif. And back in 2010, 2011, we worked with Peter and Jason, my student and I, Bruno, to try to design protein scaffolds to stabilize this helix-turn-helix and then use them as an immunogen to try to elicit motivizumab-like antibodies. And long story short, this effort did not induce neutralizing antibodies, and it was incredibly frustrating because it was, I've skipped over a lot of other attempts to design scaffolds to elicit neutralizing antibodies against HIV that we had previously done. And at this point, we were really hoping that this would be an easier test case and would provide proof of principle. But one of the things so we did design pretty nifty little scaffolds, and we had crystal structures of the scaffold bound to motivizumab. But one of the things that made us think that maybe these weren't the greatest immunogens ever was that our scaffolds did not recapitulate the full binding affinity to motivizumab that the RSVF glycoprotein itself has. And so RSVF, in its interaction with MOTA in a monovalent interaction, has a dissociation constant of 30 picomolar and a very, very slow off rate. You can really hardly measure it by SPR. And our best scaffold at this time had a KD of 90 nanomolar, so 3,000 times worse. And you can see the off rate was very, very fast. So while we were excited to be trying this proof of principle, my student and I thought, we probably can make better scaffolds that bind better. We thought maybe these scaffolds were not stabilizing the epitope conformation sufficiently. So we set out with a new strategy. We devised a new technique for scaffolding epitopes using some of that folding technology that David told you about. In fact, the idea for this method, David and I had talked about a lot when I was a postdoc, and Bruno finally coded it up. And the idea here is we want to stabilize a helix-turn-helix. We can pick a target protein topology that will be able to stabilize that conformation. And we chose a 3-helix bundle because we had some in the lab that we knew we could make. And in Rosetta, we devised software inside Rosetta that would fix the conformation of the epitope and then fold a 3-helix bundle protein around the epitope, trying to precisely stabilize the relative orientation of those 2 helices. And so this is one trajectory. It starts off in centroid mode where we're not actually simulating all the atoms, just the backbone and centroids for each side chain. You can see the epitope is frozen, and Rosetta is converging on a low resolution backbone. Now we bring in all the side chains and design and do small refinements. And so we did this, Rosetta ran through these trajectories hundreds of thousands of times, winnowing down to the ones that scored better by various filters. And we ended up picking 8 proteins to make in the lab in E. coli. And 7 of the 8 actually expressed in E. coli, and 6 of them bound really tightly to modivizumab. And this is just data for one of them. You can see by light scattering and size exclusion chromatography that it's a nice monomer at the right molecular weight. By circular dichrosum, it had a melting temperature of about 76 degrees. By 2D NMR, the dispersion was quite good. It looked like a nice protein in solution. But our favorite feature, besides all those, was in the SPR data of binding to modivizumab, you see right away this very slow off rate. And in fact, this protein has a KD of 30 picomolar, exactly the same KD as RSVF. So when we had this, we thought, all right, we can try again. We can see maybe if we improve the structure, if we improve the binding, maybe we can induce neutralizing antibodies. So this is just a table of a whole bunch of different proteins that we designed, and you can see that they're all quite thermally stable. They're all monomers, and the worst affinity that we had, I think, was 800 picomolar. So we're in a totally different realm with this new scaffolding method than we were with our previous method. To prove to ourselves that the high affinity was really because we had frozen the right conformation of the epitope, we worked with Roland Strong's lab to solve crystal structures of an unliganded scaffold and of a scaffold bound to modivizumab. And I've shown them here. Both of them are the experimental crystal structure is superimposed on the design model, and you can hardly see the difference. And that's because the overall RMSDs are really low, about 1 to 2 angstroms. And in the yellow is where the helix-turn-helix epitope is, and there we really nailed the conformation of the epitope with RMSDs at around a half of an angstrom. And it's really important that the unliganded structure has this mimicry, because that's what the immune system will see. And one other nice point about the complex crystal structure is that it did show us that there weren't any extra unintended contacts that could be helping that slow off rate. So it really was stabilizing the epitope conformation to get that high affinity. So before we immunized, we wanted to give ourselves the best shot. And David mentioned particles, and we were doing this sort of a long time ago before he had the ability to make those beautiful de novo particles. So we used a well-defined particle vaccine system, hepatitis B core antigen, and we used heterobifunctional chemical conjugation of our scaffolds to these particles, and we found that we could, on this particle, that when you express it the way we do, there are 120 of these immunodominant spikes, and we found that when we conjugated our scaffolds to these particles, we could get about 90 scaffolds per particle, which we were actually quite impressed with as far as conjugation efficiency. And we worked with Phil Johnson at Children's Hospital of Philadelphia to immunize rhesus macaques, 4 different groups of rhesus macaques with 3 different monomeric scaffolds, 001, 005, and 007, and then our 001 scaffold conjugated to these hepatitis B core antigen particles. And all of the groups made neutralizing antibody responses, which was the first time we had ever seen a protein scaffold induce a neutralizing antibody response. So we were incredibly happy about that and encouraged. And the particle group had the group averages are shown here, and the overall titers were higher in the particle group. And this paper is basically accepted for publication. And one of the reviewers said, you know, your neutralizing titers are encouraging, but I want to know how they compare to a number that I understand. And so we worked with James Crow and also Barney Graham, who had neutralization assays and also collections of human sera that were immune to RSV. So we could compare in the same assays side by side the neutralization titers from humans to the neutralization titers that we induced by vaccination. And this data is from Barney Graham's assay. And so he had 20 different humans in his assay. The mean titer was something like, I think this is 500. And The two of the 4 animals in our particle group that responded the best are shown here. The other one is down here, and the 4th one didn't really make neutralizing antibodies. But you can see that these 2 animals are responding right in the same realm as immune humans. And so that was really encouraging, especially considering that these titers are derived from natural infection, where you're exposed to F protein and G protein and multiple different neutralization epitopes, whereas these titers are derived from just a single structurally stabilized epitope. So we wanted to understand more about why we were getting this neutralizing activity and prove to ourselves that it was directly through the epitope. So we worked with Rich Wyatt, a neighbor of mine, to isolate monoclonal antibodies from one rhesus macaque that was immunized that was making potent neutralizing activity. And we recovered a bunch of monoclonals using B-cell sorting. Most of them actually didn't bind to RSV F protein. They did bind to the scaffold, but not to F. But 2 of them did bind to RSV F. And they actually are about 10 times more potent at neutralizing RSV than Synagis. So these 2 antibodies run side by side in a microneutralization assay against Synagis. And so you can see here's the IC50 for Synagis, and these guys are shifted over to lower concentrations. So that sort of agreed with the high potency of neutralization in the sera in that animal. Another really interesting thing about these monoclonals is that they have incredibly high affinity for the eliciting antigen. By SPR, both of these antibodies bind to the scaffold with an affinity of 3 picomolar. And so we're interested in that because maybe if you give a very rigid epitope, it's easier for an immune system when it's sort of basically screening its library and undergoing somatic hypermutation. Maybe it's easier to reach very high affinity in a rapid manner. And this is probably my favorite slide about this story. We solved crystals, Roland Strong's lab solved crystal structures of these monoclonals bound to the scaffolds. And so just for comparison, here's the crystal structure of modivizumab bound to the epitope in yellow. And here's a crystal structure of one of our rhesus macaque monoclonal antibodies bound to the epitope in yellow. And then on the far right, we have superimposed the 2 yellow structures and shown what the side chain conformations are. And you can see that the macaque antibody that we elicited by vaccination using our scaffold, it binds to an epitope structure that is basically identical to the epitope structure that we designed the scaffold on in the first place. The backbone is the same, and almost all the side chains are incredibly similar. And then we've colored orange side chains that are buried by one antibody or the other. And so places that have 2 side chains that are orange mean both antibodies bind that side chain, engage that side chain. And so 85% of the buried side chains are shared. And so this is really precise re-elicitation. The antibody is genetically completely unrelated to modivizumab. And it comes in at a different angle of approach, but it engages exactly the same epitope. And this is what we think we need to do for HIV, basically. So this is the first example of closing the loop of reverse vaccine engineering, where you start with a neutralizing antibody. We're given crystal structures of the epitope with and without an antibody, or actually in this case only with. We pursued epitope-focused vaccine design, biophysical analysis, structural analysis to try to select our best antigens. We immunized and we successfully re-induced neutralizing antibody against the exact same epitope. And so I think I've gone, so basically this is a proof of concept for structure-based epitope-focused vaccine design. Artificial scaffold immunogens can induce potent neutralizing responses. The induced antibodies can have very high affinity and complementary fine specificity. I didn't go into that. We think the data shows that epitope structural mimicry and antibody affinity in the antigen itself are important for elicitation. Multimeric presentation is important. The titers in the particle group were better than the monomer group. Most importantly is this idea that the fact that we could recapitulate the mode of neutralization specificity establishes feasibility for the type of immunofocusing that we think we need to do for HIV, influenza, and even for dengue and some other targets. So I'm going to quickly tell my next story, which is fairly different. So back to trimer structure. And now we're going to talk about the CD4 binding site, this yellow patch. And just to remind you, I showed you this movie, and this is the patch that we're talking about, and we need to get antibodies against that, that little back of that cave. And there are antibodies that are broadly neutralizing and bind to that yellow patch, and they fall into two classes. One class are sort of standard kinds of antibodies that rely on their CDRs to engage the epitope, and we're calling them loop-reliant in this slide. B12 was an early one that Dennis discovered. Chavi 103 is a newer one that Bart Haynes has discovered. But there's another class of antibodies that's pretty unusual that rely very strongly on their heavy chain framework to make contacts with antigens. So here, GP120 is green, and this is VRC01, and in this bluish color is the VH gene. And these balls are, all but one of them are germline encoded residues. And so it provides a really interesting opportunity for vaccine design, because everyone in this room, we think, is making plausible precursors to this broadly neutralizing antibody, and if we could figure out how to activate these precursors and drive the right amount of somatic mutation, we might be able to induce them. So just to, so this is GP120 with CD4 bound to it in orange, and there's a little movie to show you. This is VRC01, the FV portion, showing how well the heavy chain mimics the binding of CD4. But then there's also this, the light chain. Let's see if I can do this. Okay. It's failing. Okay, yeah. So now CD4 is the dark blue, and the space fill is the heavy chain of VR. It's the other way around. The space fill, I think, is CD4, and the dark blue you can see is taking up the space of CD4. The heavy chain is taking up the space of CD4. But then there's this light chain hanging off to the side, and this is the problem that the human immune system confronts when it's trying to squeeze a Fab into that CD4 binding site. The functional constraints on HIV are that it needs to bind to CD4, which is a single IG domain, and the immune system needs to squeeze a Fab in there, which is twice as fat. And so how does HIV deal with the light chain? I don't know why it's... So we wanted to study what the requirements for broad neutralization are for these antibodies. And one of the problems with these antibodies is that they're incredibly highly somatically mutated. And this slide is showing VRC01 with the heavy chain and the light chain. And in pink balls are all the locations of somatic mutations from germline. It's 30% mutated from germline. I don't know why this is not being helpful. Sorry about this. And we used, David mentioned yeast display, and Dane is here. My student screened libraries of VRC01FV on the surface of yeast, in which in the library, at each position, we allowed either the germline or the mature or the mutated residue. And we found that we could winnow down the mutations to just 9% and still have a broadly neutralizing antibody. It's just pausing on me. I'm sorry about this. So we have, in the minimal mutated VRC01, we have some mutations on the heavy chain in red and some mutations on the light chain in blue. And we actually have a crystal structure showing that these mutations on the light chain are really all to deal with this glycan that's present at the edge of the CD4 binding site. And it's revealing that what we thought before was an antibody that just slid into the CD4 binding site and really didn't have to deal with the glycans. It actually turns out that about 1/3 of the surface area that this antibody binds to, it involves this glycan. So it's actually not too terribly different from the antibodies that engage glycans at the top of the spike. And this data at the bottom shows, this is data from Devin Sock and Dennis's lab, that this min VRC01 that's only 9% mutated is basically just as broad and potent as VRC01 itself. And so in one way, we're trying to lower the bar for a vaccine, because if you think you need to design a vaccine against this specific epitope and you think it needs to be a 30% mutated from germline antibody in order to be broadly neutralizing, that's an incredibly tall order. And what we're showing is that, well, we think maybe we only need 9% mutation. And in a real vaccine case, we might get additional mutations that come along for the ride, so maybe we need 18% or something like that. But it's not as bad as 30%. And the last part of the story involves activating this germline. So what had been shown, and we published a paper about this earlier this year, so I'm just going to go quickly through it. What had been shown before our work was that all the different members of the VRC01 class of antibodies bound very, very tightly to HIV envelope, but in many different cases, if you produced a predicted germline precursor for one of these antibodies, you couldn't detect any binding to the HIV envelope protein. And the problems, we believe, are both in the heavy chain, in the inability to engage GP120 CD4 binding site productively with the heavy chain, and also in the light chain, there are problems with the germline antibody actually clashing with this glycan at 276. Right, so this circle shows where we need to fix this problem, and there are these clashes. And so we set out to engineer an artificial immunogen that would have an artificial CD4 binding site that would get around those problems. And first of all, we deleted that glycan. But we also had to make several other mutations in an already engineered molecule. So this EOD is called engineered outer domain that was already working in my lab as a minimal representation of the CD4 binding site. And on top of that, my student Joe Jardine engineered these other mutations to improve germline binding to germline precursors of VRC01 antibodies. And that was also using yeast display screening of computation-guided libraries. And in fact, this is a crystal structure of our EOD GT6 bound to germline VRC01. And so I mentioned that the EOD was previously in my lab, and it already bound very well to mature versions of all these antibodies are in the VRC01 class. They all derive from the same VH12 heavy chain, but they have different light chains and different CDRH3s. And this engineered outer domain bound very well to all of the mature antibodies, but it didn't have any detectable affinity for the germline versions, as wild-type envelope didn't either. But then when Joe engineered these germline targeting mutations in this molecule EOD-GT6, now we do have a very good affinity for germline VRC01 and several other members of this class. So the idea is that because the VH12 gene is used in about 1% of all human antibodies, and we've looked at what the other requirements are for an antibody to be a potential precursor to make a VRC01 class response, we think that we have something like a 1 in a million, maybe 1 in 10 million chance of activating in any one person at any one time if we have an immunogen with appreciable affinity for the germline. So to make a better immunogen, we similar to what David talked about, in this case we didn't do fancy computational design, we just did genetic fusion. We engineered self-assembling nanoparticles that displayed 60 copies of our engineered outer domain with the germline targeting, and these are fully glycosylated as shown in that model, and we have EM data showing what they look like. And then we worked with David Namazi at Scripps to do B-cell activation assays. And so if you have B cells that are expressing mature VRC01 IgM on their surface, both nanoparticles that were displaying the regular EOD and also the germline targeting EOD, both were capable of activating these B cells very potently. But B cells that were displaying germline VRC01 could only be activated by the germline targeting nanoparticle. And so I think this is second to last slide. The idea that we're pursuing, or one of the ideas that we're pursuing, is that we would use these GT6 nanoparticles to activate a germline B cell and drive some initial somatic mutation. And some of those antibodies are going to go in the wrong direction, but hopefully some of them are going toward an antibody that can bind to wild-type envelope. But we think that one thing we may need to do is, before we give wild-type envelope like that trimer as a boost, we think we need to shepherd the somatic mutation a little bit further. And so our second immunogen, one idea is to come in, say, with an envelope trimer that lacks that glycan, so that we can drive the appropriate mutation on the heavy chain and give the light chain a break, and then come in again, finally, with a wild type envelope to drive further the somatic mutation on the light chain. Now, of course, we can dream up all kinds of scenarios, and basically we need to start testing them in the lab. And we're working with 2 different groups that are making knock-in mice that express germline precursors to these antibodies, and we're talking to different companies that have mice with human repertoires. And I think it's going to be really exciting to try out these types of scenarios and see how can we manipulate, first of all, can we activate the right germline genes, and secondly, if we can we direct the somatic mutation to reach these broadly neutralizing antibodies. So I hope that I've showed you that a key challenge for HIV vaccine design is immunofocusing to broadly neutralizing antibody epitopes. And I didn't go over it, but it's true that there are similar challenges for other antigenically highly variable pathogens like influenza, if you want to make a universal flu vaccine, hepatitis C virus, and there are also related challenges for dengue. Even though it's not as antigenically diverse, it's incredibly tricky to make a vaccine for dengue, and we think that epitope-focused approaches might be a way to induce potent type-specific neutralizing antibodies that won't enhance infection by other strains. In the 2 stories I showed you, the RSV scaffold immunogens, the recapitulation of the mode of neutralization specificity provides proof of principle that epitope-focused vaccine design can provide can achieve the immunofocusing that we need with high precision. It doesn't mean that we'll be able to solve all these problems, but at least we feel like we're in the ballpark. The RSV scaffolds themselves actually have promise for further development as a human RSV vaccine for infants. It's sort of interesting, failed clinical trials in the 1960s of an RSV formalin inactivated virus actually led to the death of led to enhanced infection and the death of a few vaccinees. So there's incredible hesitancy on the part of the FDA and everybody else in the field to immunize infants with anything resembling RSV if it's not a replicating virus. And these scaffolds are so different from RSV, they may have a lower safety barrier to getting into a human clinical trial in infants. And finally, vaccines to induce VRC01 class broadly neutralizing antibodies against the CD4 binding site. I hope I showed you that combining germline targeting and our definition of the minimal somatic mutation requirements for this class of antibodies suggest a novel structure-based pathway for eliciting these kinds of antibodies. So I'd just like to thank, there's a whole bunch of people that contributed to the work. Let me just start by thanking David and Dennis, and I mentioned them at the beginning. You know, I really learned structural biology and computational design from David, and while I was in his lab, I was collaborating and learning from Dennis, and now I'm basically in the same building and continue collaborating and learning. So that's really great to sort of bring these 2 guys together in a way. I'm sort of a double spin-off. The RSV work was done by a student in my lab, Bruno Correa. The germline targeting was done by Joe Jardine. The nanoparticles were done by Sergei Menes, and Po-Su Huang is now a postdoc in David's lab, but he developed the EOD in the first place, and a lot of other people have contributed. I just want to call out to Phil Johnson and James Crow for collaborating on RSV. Roland did the crystallography on RSV, which, as you saw, is really essential to our understanding what we achieved. Ian Wilson's lab did the crystallography on the EOD GT6. And David, and I mentioned David Namazi for B cells and Rich Wyatt for isolating antibodies. Thank you.
Thanks very much, Bill. Very nice application of computational design. Any questions? So how many, do you think you can apply this to all viruses? Is this going to be applicable to everything?
Well, I mean, the nice thing about vaccines is that what we know is that the correlative protection for almost all licensed vaccines is eliciting functional antibodies that neutralize or fix complement or cause opsonophagocytosis, and antibodies recognize structure. So in that way, it should be very generic. So we think it's very generalizable. What we need is someone to find protective monoclonal antibodies that are accessible by the human repertoire, and we need structural information about what their epitopes look like and what the steric environment around that epitope looks like.
And then the second question is, with respect to the idea of educating the immune response to go from a germline to a final neutralizing antibody seems to me like the order and time sort of scale of giving these individual vaccines is going to be really crucial. So there's no missing a dose in the middle. You've got to do it properly, otherwise, or do you think that's not going to be an issue?
Yeah, I mean, definitely, you know, all the aspects of the regimen, the timing and the dose and you know, all those details, we just have to test them empirically. I think we can hypothesize about optimal situations, but there's basically zero data on this kind of thing to try to do this, so we need to do a lot of experiments.
A couple of questions at the back there.
Yeah, I have two questions. The first question, do you think the similar approach can be applied not just for the vaccines or infectious diseases, but also, you know, GPCR or ion channel types of targets where often the structure seems to be important in generating antibodies?
Yeah, so we're super interested in that, and we're talking to some people about collaborating on those kinds of applications. I think there are lots of applications there. One can imagine designing antibodies for those things, but we're also really interested in stabilizing the appropriate antigenic structures so that antibodies can be pulled out of libraries or obtained by immunization.
Okay. The second question is more like technical. You mentioned you looked for the IgM positive B cells activation by the nanoparticles. What's your readout there?
Oh, that assay was calcium flux.
Okay, thank you.
Great talk. In some earlier studies of a stem vaccine-like construct, I think the observation was that the serological memory declined quite rapidly after a few weeks. And I was wondering if you have any longer-term data on serological responses and whether you have neutralizing antibodies that are maintained?
So you mean in the macaques that we
in the
immunize?
macaques, yeah, in this particular case. Yeah.
Well, what we do know is that the neutralizing titers were, they peaked after the 5th immunization at 20 weeks, and by 32 weeks the titers were waning already. We haven't measured beyond that, but it doesn't look like we've solved the problem of durability at all. And for HIV, I talked a lot about the specificity of the response that we need, but that even if we achieve that, that won't be a vaccine. I mean, I'm skipping past a lot of other details, and one of them certainly is the durability. For HIV vaccine, we need to maintain high titers of circulating IgG permanently in an individual, and how we do that is, another question.
Thank you.
Okay, if that's it, then we'll break for lunch.